Hello everyone and welcome to yet another recorded lecture of BIO1 online. Congratulations on making it through the first unit so far. Today we will start the second unit, which begins with chapter 7, which is on DNA structure and gene function. So what is DNA? We said way back at the beginning that DNA is a molecule of nucleic acid. Specifically, DNA stands for deoxyribonucleic acid. And like all nucleic acids, DNA is made up of many monomer subunits called nucleotides. Right? DNA is a polymer of many monomers of nucleotides. And DNA can store the information that the cell needs to produce proteins. So DNA is an information storage molecule. But scientists didn't know that for quite some time. And I want to just talk about this brief experiment that Griffith did. He was an English microbiologist. This was done in the late 20s. And what he did was he took two separate strains of bacteria. One was called the R strain and one was called the S strain. And when the R strain was injected into a mouse, the mouse was fine. But the S strain was lethal. So something about the S strain was pathogenic, was harmful, but something about the R strain was not harmful. But when they took that harmful S strain and killed it with heat, the mouse was perfectly healthy. So this showed that something right in the S strand could be killed. But this is where it got interesting. When they took the heat-killed S strain and the living R strain that was otherwise harmless, the mouse still died. So Griffith's experiment showed that some identified substance in this heat-killed strain S was able to transform this harmless strain R and still kill the mouse. Not only that, live strain S bacteria was retrieved from the dead mouse. So clearly, something was being transmitted from the heat-killed S strain into the R strain. And later, it was discovered that DNA was actually transmitted between the toxic S strain and the harmless R strain. So later, we knew that DNA was this transforming substance, but that wasn't very clear, even at this point. They just knew that something was being transmitted between this heat-killed strain and this harmless strain. Hershey and Chase, some years later, were able to figure this out. So it was in the 50s where Hershey and Chase showed that it was DNA and not protein that contained genetic information. And they did a very simple experiment in order to illustrate this. So they used E. coli, which is a type of bacteria, for their experiment, and they used a special kind of virus called a bacteriophage that can only infect bacteria. So this is a virus that infects bacteria. The virus has two major parts to it. It has a protein coat and DNA on the inside. So what they were trying to figure out was that does the virus insert its protein and replicate its proteins in the bacteria, or does it insert its DNA and replicate the DNA in the bacteria? That was what they were trying to answer. So they took two different batches of bacteriophage. One in which the protein coat was radioactively labeled. So again, sulfur specifically. And the protein was radioactively labeled, so the protein would glow, so to speak. And then in a different batch, they radioactively labeled the DNA. They then took two separate batches of E. coli and infected them with each of these radioactively labeled viruses. After infection, they blended the viruses and the bacteria and spun them down at very high speeds to separate all the bacteria, which would sink to the bottom, and then all the excess virus. So they did that with both. And remember that the bacteria cells would be heavier, so they would kind of sink down when you spin them in a centrifuge. And what they wanted to look at was what is inside the bacteria, right? Is it radioactive DNA or is it radioactive protein? And what they saw was that with the cells that were infected with the protein coat labeled, there was nothing radioactive inside the bacteria. However, when the DNA was radioactively labeled, they saw all the bacteria had radioactively labeled viral DNA. So this was proof that it's DNA that is the transforming principle that can go from a virus into a cell that can actually change their properties. So DNA is a nucleic acid built of deoxyribonucleotides, we said, and each DNA nucleotide has 3 parts. It has a sugar. In the case of DNA, it's always deoxyribose. It has a phosphate group, and it has a base. We say this base is nitrogenous because it has many nitrogens. The base can be one of 4 types. You can have an adenine, which we abbreviate A. We have thymine, we have guanine, or cytosine. So which is not part of a nucleotide? Pause here and answer this question now. And you know the answer is B. A lipid is not part of a nucleotide. So DNA is double-stranded. So what that means is a double-stranded DNA molecule has 2 separate strands, 2 separate molecules. So a DNA molecule is a bunch of nucleotides attached together. So nucleotides join to form strands, right? And DNA is double-stranded, so that means we have 2 separate strands. Right? DNA is double-stranded. The sugars and the phosphates of each nucleotide form what's called the DNA backbone. So look over here. Here's one nucleotide. And it's the phosphates and the sugars that stack up to form the sugar-phosphate backbone. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. The bases from each DNA strand stick out. So it's almost like a ladder, right? And the steps of the ladder are the bases. The bases of each strand of DNA are joined by hydrogen bonds. So these 2 strands of DNA are not physically attached. They're very weakly attached by many hydrogen bonds. And what's very important about DNA is that base pairing is complementary. Complementary with an E, not like a compliment that you're being nice to somebody, but complementing meaning they match. Specifically, if there is an adenine on one strand of DNA, it can only make hydrogen bonds with a thymine on the other strand. If there is a cytosine on one strand of DNA, it can only make hydrogen bonds with a guanine on another strand. So that's very important. And again, notice how in this picture we have 2 strands of DNA. The sugar-phosphate backbone is shown in red. The bases are sticking out. Hydrogen bonds are shown by these dotted lines. But these can be separated. With heat, hydrogen bonds break very easily. We said there are 4 nitrogenous bases. Adenine and guanine are called purines. Purines, you can remember that anything that ends in -nine, N-I-N-E, is a purine. And purines have 2 rings to it. So here's an adenine, it has 2 rings, 1, 2. And here's guanine, 1, 2 rings. Thymine and cytosine are called pyrimidine bases because they only have 1 single ring. There's thymine, just 1 single ring. And a purine can only match up with a pyrimidine, right? A with T, G with C. Those are base pairing rules. Chargaff's rule. Chargaff was a scientist that discovered or helped discover base pairing. And he basically states that the amount of adenine will equal the amount of thymine, and the amount of guanine will equal the amount of cytosine because of this base pairing rules, right? So all of this information suggests that DNA uses complementary base pairing to store genetic information. And this gives us a great number of possible nucleotide sequences, right? With just 4 different letters. Since we have so many different instructions, we have lots of possibilities of different nucleotide sequences. So which of the following is not a DNA base? Using process of elimination, you should figure out that it is C, uracil, right? It's not a DNA base. So we said that DNA kind of looks like a ladder when it's in a double-stranded form. Really, it's like a twisted ladder, right? It doesn't really look like this flat. It's a twisted ladder. And each strand of DNA is not in the same direction. What that means is that each strand of DNA has polarity. Each end is not like the other. The ends of each strand are unequal. Right? That's what polar means. It's unequal. Specifically, the end with the exposed phosphate is called the 5' end. And just so you know, it's because this is the 5th carbon in the sugar. And that's why the phosphate's attached to the fifth carbon in the sugar. That's a side point. But for now, you should know that the end with the phosphate sticking out is called the 5' end. Right? It's attached to the fifth carbon of the deoxyribose. The other end of the strand is called the 3' end. So this is the unbound end. This is where the hydroxyl group is attached to the third carbon of deoxyribose. So specifically, this is the first carbon, second carbon, third carbon. That's where the OH would be. Right? Fourth carbon, fifth carbon. So the phosphate's attached to the fifth carbon. The OH is always attached to the third carbon. And that's why we just call it the 3' end. That's what you need to know, is that the end with the phosphate sticking out is the 5' end, which means the other end is 3'. And the critical point of this is to show that the two strands of DNA are anti-parallel to each other. One strand goes from 5' to 3', the other goes from 3' to 5'. Right? So one goes in this direction, the other goes in this direction. You can never have two 5' to 3' going in the same direction. Will never happen. You're never going to see 5' to 3' on top of a 5' to 3'. Cannot be. X. No. You can only have 5' to 3' anti-parallel with another 5' to 3'. And this is another very important rule I want you to remember, is that genes are only read from 5' to 3'. So gene sequences are read 5' to 3', just like sentences are read starting with the capital letter and ending with a period. So this gene sequence could be 5' A, G, T, C, 3', or 5' G, A, C, T, 3'. We always read genes 5 to 3. This is just another schematic showing the directionality of DNA. So we said that the two strands of a DNA molecule are complementary to each other, right? The sequence of one strand determines the sequence on the other strand. And we said that these two strands are anti-parallel to each other. In general, when you're writing gene sequences, you always want to write 5' and 3'. Just like when you punctuate your sentences, you want to write 5' at the start of your gene sequence and a 3' at the end of your gene sequence. What type of bond holds two DNA strands together? So pause here. And the answer is hydrogen bonds hold DNA strands together. Remember, hydrogen bonds are rather weak on their own, but because of the specific structures of the complementary bases, they can maximize tight binding with each other, because a DNA molecule consists of millions of base pairs. So it has millions of hydrogen bonds. So collectively, those bonds are strong enough to hold the two strands together, yet weak enough to pull them apart when the cell needs to use its DNA. So hydrogen bonds are very critical to the structure of DNA. Now let's talk a little bit about DNA structure. And a lot of what we know about DNA structure is thanks to Rosalind Franklin, a female who studied the structure of DNA using X-rays in the 50s. And being a female and being a Jew, she actually had the odds stacked against her, but she made very, very notable contributions to biology. Under the right conditions, DNA can produce what's called an X-ray diffraction pattern. If you crystallize DNA under certain conditions, you can use an X-ray beam, and the X-ray will hit the atoms of DNA in such a way and produce a pattern. A way to kind of illustrate this or to imagine this, it's kind of like if you were to drop a pebble into a pond, and you could look at the ripples. And then from looking at the ripples, you can deduce the exact shape of that rock that you dropped. That's the same idea. So by looking at this diffraction pattern, she looked at a lot of different photographs of crystallized DNA. She provided evidence that DNA had certain features. She proved with this one photograph, this is a very famous photograph, that DNA is a helix, and that the helix continues to repeat over and over again. So Rosalind Franklin's X-ray diffraction technique helped determine the 3D shape of the molecule. And a colleague of hers called Wilkins showed her photograph to James Watson, who understood its significance about DNA structure. And taken together, Watson and Crick, who are both, Watson was American, Crick was English, and they worked together and solved the mystery using Chargaff's rule and Franklin's X-ray diffraction pattern. And they showed that DNA is a double-stranded helix, like a twisted ladder, that go in anti-parallel directions. Right, the two chains run in opposite directions, and the bases form horizontal connections between these two backbones. So this was huge, and Watson, Crick, and Wilkins won the 1962 Nobel Prize for their discovery. So just to review, we said DNA has a sugar-phosphate backbone. Complementary base pairs in the middle are joined by hydrogen bonds. Anti-parallel strands means that one goes 5 to 3, the other goes 5 to 3 upward. And when it's twisted, it forms this double helix. So this twisted form is actually because of hydrogen bonds between the backbone. So there's some weak interactions between the sugars and the phosphates in the DNA backbone that allows it to form this three-dimensional double helix conformation. And DNA is most stable in this form. So stable that I can go to ancient Egypt and take DNA samples from a mummy that has been decomposing for thousands of years, and the DNA is still intact. Right? And again, the structure, that twisting structure, is due to hydrogen bonds in the backbone. So hydrogen bonds are between the bases, but also in the backbone. And you can pause here just to get a summary of how DNA was discovered. Let's watch this video together to summarize what we know about DNA so far.
The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands.
Now let's discuss genes and genomes. So a gene is a sequence of DNA that contains biological information. Usually the biological information are instructions to make a protein. So a gene is usually a sequence of DNA molecules that encodes a specific protein or an RNA molecule. Genes are spread out in long DNA molecules called chromosomes. So we have 23 pairs of chromosomes. We got 23 chromosomes from mom, 23 chromosomes from dad, giving us 46. And each set of 23 chromosome pairs, or each set of 23 chromosomes, has 20 to 30,000 genes that code for proteins. So the human genome includes 20 to 30,000 genes scattered on 23 pairs of chromosomes. And the genome is all the genetic information of the organism. So an organism's genome is all of the genetic material. And genomes vary in size and complexity, and usually most of the genetic information of an organism does not code for proteins. We actually have yet to understand a lot of the sequences of DNA. But we do know from the Human Genome Project many of the genes that code for proteins. So the Human Genome Project kind of looked inside the human user manual, right? What instructions make which kinds of proteins? Almost like a cookbook has recipes for different dishes, right? The genome has different instructions for different proteins. Right? And we said the genome is stored in the nucleus, right, as chromosomes. And if we were to unwrap these chromosomes, we'd see a double helix of DNA wrapped around histone proteins. And those genes are short segments of genetic instructions interspersed amongst these chromosome pairs. Right, so you can pause here to get a sense of the Human Genome Project, which helped us figure out which genes coded for which proteins, and also helped us understand how misspellings in the sequence or mutations can lead to disease. So where is DNA stored? Well, in prokaryotes, DNA is stored in the cytoplasm. And prokaryotes, like this E. coli bacterium, has a single circular chromosome. It's found in what's called the nucleoid region, but it's not a membrane enclosed nucleus, right? Prokaryotes do not have organelles like a nucleus. In eukaryotes, DNA is stored in the nucleus. And eukaryotes may have multiple linear chromosomes made of chromatin. So the human genome looks something like this. We have 23 chromosomes, right? 23 chromosomes as shown here. You can have either an X or a Y from each set that you get from your parents. And we said genes are interspersed among chromosomes. All right, so we have many chromosomes with 20 to 30,000 genes spread amongst them. So you can pause here and answer this question. And the answer is B, the nucleus. This is a little, well, I don't want to say it's tricky, but you should know that mitochondria and chloroplasts do have their own DNA. But those code for mitochondrial proteins and chloroplast proteins, not the proteins needed for our functioning of every cell in our body. So the human genome is contained in the nucleus, but DNA is also found in the mitochondria of human cells. Where is the E. coli genome contained? So pause here. And this answer is specifically A, the cytoplasm. So like I said before, humans have 23 pairs of chromosomes. We get one set of 23 chromosomes from mom, one set of 23 chromosomes from dad. So this is a lot of information we have in each of our cells. So in every single one of your cells, you have 20 to 30,000 genes. But again, that's only like 1% of the genome codes for proteins. So your genome is incredibly large. And to illustrate this, let's break it down. Each cell has 2 meters of DNA. So one cell, which is smaller than a pinpoint, has 2 meters of DNA. That's 6 feet. That's bigger than you. The human body has way over 10 trillion cells, right? Up to 40 trillion cells. So if you multiply 2 meters of DNA per cell times 10 trillion cells, that means the length of the genome in the human body is over 20 billion kilometers. And just as a comparison, the distance from the sun to planet Neptune is less than 5 billion kilometers. So you could go from the sun to Neptune back twice, 2 round trips from the sun to Neptune, and that's the amount of DNA in your body. If the DNA in one of your cells were typed out, if I just typed out your human genome on regular paper, size 12 font, Times New Roman, normal margins, and I stacked up all the pages, that stack would be almost as high as the Washington Monument and taller than the Statue of Liberty and Big Ben. It would be about 420 feet high, just your genome typed up in regular size 12 font. So clearly, we have a lot of information in each of our nuclei. So how can all of that DNA fit inside a tiny cell? Right? We have all this loose thread. But just like we don't store loose thread in our sewing kits, right? We have spools to wrap our thread around to organize it. And the same thing happens in the genome, right? We have protein spools called histones. Histones are groups of 8 proteins themselves that act as spools to wrap DNA around. And this could help condense DNA about 10,000-fold. We call DNA wrapped around histone proteins chromatin. Chromatin. So chromosomes are made up of chromatin. Chromatin equals DNA wrapped around histones. So DNA is always in the form of chromatin, and it's referred to as beads on a string because it looks like the string is DNA and the beads are those histones. And one histone octamer with DNA wrapped around it is called a nucleosome. So here's a nucleosome, here's another nucleosome, nucleosome. And again, this is all in an effort to take that double helix, which is so long and has so much information, and wrap it and continue to wrap it so all of that DNA could be condensed and fit inside of each of our cells. So that's a little talk about DNA for now, but now we're going to start talking about RNA. And RNA is a close chemical cousin to DNA, but with some key differences. Whereas in DNA, the sugar is deoxyribose, in RNA, the sugar is ribose. And that just has an extra O over here. This is deoxyribose. It has one less oxygen. We said DNA has A, G, C, and T, where adenine and thymine are complementary to each other. But in RNA, there is no such thing as thymine. There's only uracil instead of thymine. So adenine matches up with uracil in RNA. That's important. Where DNA is usually double-stranded, RNA is generally single-stranded. And DNA is an information storage molecule that transfers information from one generation to the next. RNA has many, many functions. It also carries protein encoding information, but it can also help make proteins and catalyze reactions as an enzyme. So RNA is a very, very cool molecule. So now we can introduce the central dogma of biology. And this is as important as it sounds. So in the 40s, it was discovered that genes control the production of proteins. And in the following decades, Watson and Crick described this relationship between nucleic acids and proteins as the flow of information called the central dogma of biology. The central dogma of biology states the following. Genetic code in the form of DNA has instructions to make proteins. The DNA instructions get transcribed or rewritten into an RNA message first, and then the ribosomes make proteins using that RNA message. That's what the central dogma of biology states. In other words, the mechanism of how genes are expressed is that the DNA in genes specifies information, but the information is just digital, meaning it's made up of A's, T's, C's, and G's. The information is not structural or functional. You can't use DNA to do anything in the cell. It just has information, right? Just like a cookbook would. You could also think about it as like in coding, we could use bits like zeros and ones to program software. Well, DNA, it doesn't use zeros and ones, it uses A's, T's, C's, and G's. But those A's, T's, C's, and G's, that genetic information can be expressed into structure and function through protein synthesis. So the expression of genetic information into structure and function that could actually do something in the cell is explained by the central dogma. And this is a huge concept in biology. DNA in a gene determines the sequence of nucleotides in an RNA molecule. And it's the RNA that controls the primary structure of a protein. And remember, what the primary structure of a protein is just the linear sequence of amino acids connected by peptide bonds. So what we're going to see is that DNA determines the sequence of nucleotides in RNA, and RNA, the sequence of RNA nucleotides, determines which amino acids get put into a protein. We can divide up protein production in two stages. The first is called transcription, and that's where a cell rewrites the genetic message of DNA into a complementary RNA molecule. This RNA message can then be translated. So translation is how proteins are made using the RNA instructions. So transcription is DNA to RNA, translation is RNA to protein. We could compare this to like a cookbook is like our genome that has, or our chromosome that has many, many genes in it. But I don't want to make every single recipe in this entire cookbook. Right, that wouldn't really make sense. What I want to do is instead of taking the whole cookbook down to the kitchen, let me just make a photocopy of the brownies I want to make. So I'm going to take a photocopy of just the brownie recipe, then I'll bring that down to my kitchen. And then according to whatever the brownie recipe says, I'm going to assemble those ingredients, those amino acids, in the right sequence so I can make my protein or my brownies. Right, so again, transcription is the idea of I'm not taking my whole cookbook from the library down to the kitchen. I'm just going to make a copy of what I need. I'll take that one copy down, follow the sequence of its instructions to put together my building blocks so I can get my final product. So the central dogma says that DNA can get replicated, so DNA can become more DNA. DNA gets transcribed into RNA. RNA gets translated into proteins, which are made up of amino acids, and that happens on ribosomes. We also know that RNA can actually make more of itself, and something you don't have to know for this class, but RNA can actually be reverse transcribed back into DNA, we now know. What is the main function of DNA? So pause here, right? And the answer is A, to encode proteins. So that's the main function of DNA.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
So let's begin talking about transcription. So what's the point of transcription? Let's take a step back. In order to make proteins, we need ribosomes. But ribosomes are in the cytoplasm, right? Those are what connect the amino acids to make proteins. The DNA has the instructions on how to make the proteins, but DNA is stuck in the nucleus. So we have a problem, right? The protein producers are the cytoplasm, but the information on how to make the proteins are in the nucleus. So RNA is a close chemical cousin of DNA that can leave the nucleus. So RNA can serve as a messenger. That's why we call it mRNA, messenger RNA. So DNA is transcribed into RNA. It's rewritten into RNA so that the genetic message can be sent to the ribosomes in the cytoplasm where proteins are made. So way back in the day, you used to have a permanent record in your principal's office in high school. And if you were to apply to college, they would never send your entire record, your whole folder. They would just make a transcript. They would send a transcript or a copy, right? A message of how you performed in school, and they would send that out. They would never send the actual hard copy. They would just send a transcript. So just like you're not going to send the actual chromosome out of the nucleus, you're just going to make a copy of it into messenger RNA. And the enzyme that can do this is RNA polymerase. RNA polymerase can read the DNA code and transcribe it or rewrite it into messenger RNA. So RNA polymerase is doing that right now. So all the cells and all of your nuclei right now, any protein that needs to be made, right, has to be made from an mRNA transcript. So right now, RNA polymerase in your nucleus and all of your nuclei is rewriting some of your genetic messages into mRNA. Right? So DNA gets transcribed into mRNA by RNA polymerase inside of your nuclei. And this happens all the time. Interestingly, either of the two strands of DNA can serve as a template for an mRNA transcript. So for example, the template for gene X can be on top, but the template for gene Y can be on the bottom. So only one of the two strands contains the information encoding for a protein. We call that strand the template strand, right? And depending on the gene, the template can be either of these two. We'll see that RNA polymerase recognizes the start of a gene called the promoter, and that's what determines which of the two strands is the template. It always starts at the promoter, and I'll show you this in a bit. So we already said transcription occurs in the nucleus. RNA polymerase is the main enzyme, and since we're building more RNA, we need the building blocks. We need RNA nucleotides to form the letters of our new mRNA. And of course, we need something to read from, so we need our template DNA strand. At the end of transcription, we're going to get an mRNA copy of the DNA genetic code. There are three basic steps. There's initiation, elongation, and termination. We'll talk about each of those three now. So, step one of transcription is initiation. And this is important, so write this down. Every gene starts with a sequence of DNA called the promoter sequence. The promoter is the start to every gene. So only the template strand of a gene will have the promoter. And that's where RNA polymerase recognizes the start of a gene. So in order to transcribe a gene, RNA polymerase must bind the promoter of that gene. So here's RNA polymerase. It recognizes the beginning of this gene at the promoter. That's very important. Several other proteins called transcription factors help RNA polymerase find the right promoters. I'll talk a lot more about this later. But basically, if you're in an I cell, you have certain transcription factors telling RNA polymerase to go to all the I-related genes, because we need I proteins in an I cell. In a muscle cell, you're going to have different transcription factors telling RNA polymerase to transcribe different muscle genes. That could be a little confusing now, so we'll talk about that later. But for now, I want you to understand that RNA polymerase starts transcription by binding to the promoter. The promoter is upstream of the gene sequence. Upstream means before in genetic terms. Downstream means after. So the promoter is upstream of the gene, or you could say the gene is downstream of the promoter. Step two is elongation. An RNA polymerase will break the hydrogen bonds of the double helix, so we'll unzip the DNA helix, and we'll start adding in RNA nucleotides to complement the DNA nucleotides on the template strand. So RNA polymerase is going to come in, unzip the double helix, and if it sees a cytosine in DNA, it's going to put a guanine of RNA, right? Because C goes with G. If it sees an A in DNA, it's going to put a U of RNA. Remember, there's no such thing as T in RNA. There's U instead of T. If there's a G in the DNA, RNA polymerase will put a C, right? G with C. So you have to understand the base pairing rules to do transcription, right? Very importantly, RNA polymerase can only make mRNA in one direction. It only knows how to synthesize mRNA 5' to 3'. So specifically, this was the first, it started up here and ended over here. RNA polymerase can only make mRNA 5' to 3'. What that means is that it can only add a nucleotide to the 3' end. So in this example, the last and most recent nucleotide added was this G. Right? The first thing put in was here. So RNA can only be made 5' to 3'. New nucleotides can only be added to the 3' end. This might not be very obvious or clear at first, so pause here and try to make sure you understand this. RNA polymerase can only add RNA nucleotides to an existing 3' end. Therefore, RNA polymerase has to start at the 3' end of the DNA template and will then read that template from 3' to 5' as it makes RNA 5' to 3'. Again, this might not make sense right away, but we're going to go through it until it makes sense. RNA polymerase starts towards the 3' end, so it started reading over here, and it's going to make new RNA from 5' to 3'. So RNA polymerase started reading the gene closer to this end, which is the 3' end, started reading C, and then put G. Then C with G, G with C, and so on. And again, remember that with adenine in DNA, it matches up with a uracil in RNA. Finally, the last step is termination. And RNA polymerase encounters a terminator signal at the end of the gene, and it basically just falls off from the mRNA and detaches from the DNA. The DNA helix reforms into that double-stranded DNA, and that's the end of transcription. So every gene starts with the promoter and ends with the terminator. Now the cell produced an mRNA copy of the gene.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
So let's do some transcription practice together. Suppose you're given a double-stranded sequence of DNA. You're going to be told which is the template strand, and the template strand of DNA is usually going to be written from 3' to 5'. Right? In this class, the template strand of DNA will always be given to you 3' to 5'. So what you there have to figure out, right? You say, okay, so RNA polymerase can only make RNA from 5' to 3'. So it'll start at the 3' end of the DNA template. So suppose I'm RNA polymerase. What am I going to put down first? Well, I'm going to read a T from the template, and I know what goes with T is A. So RNA polymerase will put an A. Then it reads a C on the DNA template. What will it put there? It'll put a G. Then it sees a G in the DNA template. What will it put down? A C. So I'd like you to pause here and write down the entire sequence of RNA, of mRNA that would be transcribed from this template strand. So pause here, and hopefully what you have gotten looks like this. And what you should notice is that the resulting mRNA looks just like the strand that was the non-template strand. And for that reason, we call the other strand the coding strand of DNA. The coding strand of DNA, which is in 5' to 3', looks exactly like the mRNA transcript. There's going to be one difference, and that's if this were a T, then this would be a U. That's the only difference, is with T's and U's. So again, to recap, when you transcribe a sequence, you look for you start at the 3' end of the DNA template, and then you say, okay, is it a T? That means it's going to be an A. Is it a C? G. G with C. C with G. And this would be your mRNA transcript. So let's do one more example. I'd like for you to, and again, I'm telling you which is the template strand and which is the coding strand. So what is the mRNA code? So pause here and write this in your notes, and always indicate 5' and 3'. All right, on an exam, you would not get full credit if you don't put 5' and 3'. So pause here. And this is what you should get. Right? By reading, starting from 3', you see a T, you know there's going to be A. If there's an A, that means there has to be U. If there's C, that has to be G, ... And of course, you always want to write 5'. And again, after you do that transcription, you know that the mRNA is going to look just like the coding strand, right? The complementary strand to the template, except wherever you saw a T in the DNA coding strand, you would have a U in the RNA transcript, right? So T's get switched into U's. So not to confuse you, but if you were given a DNA coding strand from 5' to 3', and you're asked what would the mRNA transcript look like, the only thing you have to do is switch the T's to U's and write a 5' to 3'. Right? If you're given a template strand written 3' to 5', then you have to figure out what RNA polymerase would do, right? So you have to read it and say, okay, if there's a T, that means it would put an A. If there's an A, that means it would put a U. So before we move on to translation, the mRNA molecule must first be processed before it can leave the nucleus to be translated on a ribosome. So now let's talk about mRNA processing. Within a gene of DNA, there are parts that actually code for amino acids and proteins, and there are parts that intervene. The parts that actually get expressed into proteins are called exons. The parts that intervene are called introns. So expressed exons, intervening introns. So introns must be removed before the mRNA can leave the nucleus. This is called splicing. So what happens is all these introns are cut out. So only the introns remain. This is done by a special RNA enzyme called SNRPs. They cut the introns out and they seal only the exons together. So the mature mRNA should have only exons, no intervening introns. One other modification has to happen, which is a cap and tail has to protect the mRNA. So there's a cap made up of guanine, there's a G, and this helps ribosomes attach to the transcript during translation. And then a special tail has to be put on, called a poly-A tail is added to the 3' end. And that's a sequence of repeating adenine nucleotides. And the more A's that are added, the more stable the mRNA molecule, and the more protein could be made from it. So again, we said we started off with something that looked like this after transcription, but we needed to do RNA processing. Specifically, we got rid of introns, right? Those introns were spliced, and then we only sealed together the introns. We put a 5' cap and a 3' poly-A tail. So let's watch a video to summarize that.
The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly-A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation.
So the processed RNA is now a fully functional molecule. Once it's processed, and we call it the mature mRNA, right after we added the cap and tail, remove the introns, it's a mature mRNA, and that can be transported out of a nuclear pore to find a ribosome for translation. What gets added before mRNA can exit the nucleus? So pause here. And the answer is a poly-A tail, D. What gets removed? So please pause here. And the answer is introns get removed. So this is a possible quiz question. If the DNA template strand has the following sequence, what would be the nucleotide sequence of the complementary RNA molecule produced in transcription? So pause here, and you're given the DNA template strand. So pause here, and I'm going to show it for you, right? If you had RNA polymerase, where would it start? Well, you know it starts at the 3' end. So it always starts at the 3' end. And if it sees an A, what is it going to put? Well, a U. If it sees a G, what is it going to put? C. T, what is it going to put? A. G. A. 3'. That is my answer, which is D. That's what would be produced in transcription. So if that is not crystal clear, do not move forward. I would recommend rereading the book again, watch the crash course videos over again until you understand this part, because we're going to build up on this concept in the next slides. So now let's discuss translation. So in translating the genetic code, we are making proteins. The point of translation is to translate the mRNA sequence of nucleotides into amino acids. We're basically taking the language of mRNA nucleotides and translating that language into amino acids. Translation takes place on ribosomes in the cytoplasm. The ribosomes may be in the cytosol, right, floating around, or on the rough endoplasmic reticulum. That determines on the final destination of the protein. Ribosomes, whether they're in the cytosol or the rough ER, translate the information from mRNA into a chain of amino acids to become a protein. And remember that every protein is nothing more than a sequence of amino acids. The sequence of amino acids is critical for the structure of the protein, right? Because different amino acids have different properties. Some are positive, some are negative, some are polar, some are nonpolar. And the sequence in which the amino acids are connected will determine the type of interactions all the different amino acids can have with each other. And that overall will determine the protein's structure. And what do we know about structure is that it determines function. So what I want you to try to begin to understand is that genetic code contains the information on which sequence of amino acids to put together. Depending on the sequence of amino acids put together, that will determine how the protein will fold, which will determine the structure, which will overall determine what the protein can do, how it can function. Right? This is the whole gist of the central dogma of biology, that each of these 20 amino acids that all proteins are constituted from, there are letters in DNA that encode for these amino acids. So let me try to explain. We started off with a double-stranded DNA molecule. We said that the template strand gets transcribed in the nucleus by RNA polymerase to form mRNA. Once we have a 5' to 3' mRNA transcript, we can look closer and realize that every 3 letters of mRNA is a code for an amino acid. So this is critical. Every 3 letters of mRNA is a secret code for an amino acid. So the mRNA code specifies the amino acids in a protein. A codon consists of 3 consecutive mRNA nucleotides that specify a single amino acid in a protein. For example, the codon 5' AGC 3', AGC, always refers to the amino acid serine. The codon 5' GAC always refers to the amino acid aspartate. So So every 3 letters of mRNA specifies a different amino acid. And there are 20 different amino acids. And there are 3-letter abbreviations for each amino acid. So let's look at alanine, is abbreviated ALA. The 1 letter is A. And there are actually 4 different codons of mRNA that could encode for the amino acid alanine. So when you're looking at a codon table, these are all written 5' to 3'. That's the first thing to know. Codons are always written 5' to 3'. So if you had GCA, GCC, GCG, or GCU, that would refer to the amino acid alanine. There is a special codon called a start codon, AUG, that is the beginning of every single protein. This methionine, right? Methionine is the amino acid encoded by the start codon AUG. So every processed mRNA transcript will begin with the start codon AUG. That's what marks the start of translation. Right? So AUG also codes for methionine if it's in the middle of a transcript. There are 3 stop codons, UAA, UAG, and UGA, that encode for the end of a protein. The other 61 codons are for amino acids. Right, so we have 3 stop codons and 61 codons for amino acids, giving us 64 total. So this is the genetic code. Right? So for serine, you have all these different possibilities. You have 6 different combinations of nucleotides could encode for the amino acid serine, which is a polar amino acid. So the genetic code is redundant. You have multiple possibilities that can encode for one different amino acid. And in order to do translation, we need to use a codon table. And a codon table lists each codon, each 3 letters of mRNA, and its corresponding amino acid. You must know how to use a codon table for this course. Suppose I tell you to find out what amino acid the codon CAU codes for. So CAU is, in other words, 5' CAU, and this is RNA. So let's see what it codes for. We look at the first base in this column, in the left column. So since the first base is C, we're going to start here in C. The second letter is A. So now we're going to look at this top part and we're going to find the A. Okay, here it is. So now we know it's going to be in the intersection of this row and this column. The last letter is U. The third base is U. And there we have CAU encodes for the amino acid histidine. So right now, tell me what AGC encodes for. What does AGC encode for? So pause here to find AGC. You go AGC encodes for serine. So, if that's clear, we can move on to talking about 2 other kinds of RNA that are involved in translation. We already spoke about mRNA, which is the transcript of DNA code that can leave the nucleus and send a message to the ribosome to make a protein. That's what was produced in transcription. Right? So the mRNA encodes the actual amino acid sequence. The codons are found in messenger RNA. We're then going to introduce ribosomal RNA, which is a major component of ribosomes. Our RNA is actually the part that can make peptide bonds. And finally, the third type of RNA you should know about is called tRNA, for transfer RNA. And tRNA can transfer amino acids to the ribosome to bring proteins, to make proteins. So the tRNA can physically link the message of mRNA to an amino acid. So tRNAs have a very, very important role. But altogether, mRNA, tRNA, and rRNA are going to work together in translation to make proteins. What type of RNA is made during transcription? So pause here. And the answer is B, mRNA is made during transcription. So we already said that ribosomes are responsible for making proteins, and these ribosomes can be found floating in the cytosol or associated with the rough endoplasmic reticulum. Proteins that are destined for secretion out of the cell or for a specific destination in the cell are made on the ribosomes of the rough endoplasmic reticulum. Each ribosome has two subunits. It has a small subunit and a large subunit. And ribosomes are mostly ribosomal RNA. So all of the green are RNA nucleotides. The purple and blue are the proteins. Ribosomal RNA is also produced from a DNA template, but that's a little more information that I want you to know. Ribosomes are really important because they can help the three types of RNA interact with each other to build a protein. So within a ribosome, we have rRNA, tRNA, and mRNA all working together to produce proteins. And the large ribosomal subunit has enzyme activity that can actually make peptide bonds between amino acids. So this is an example of an enzyme that's RNA. And this ribosome is making peptide bonds between amino acids. That's the reaction it's catalyzing. Right? The ribosomes make peptide bonds between amino acids. And the peptide bond is created many times to produce a polypeptide, which can then fold into a three-dimensional shape and become a functional protein. Now let's talk about tRNAs. So transfer RNAs transfer amino acids to the ribosome. tRNAs resemble like a three-leaf clover, and they have two important binding sites. To one end, specifically the 3' end, there is an amino acid, and this could be one of 20 different amino acids. The other end has a very important sequence of 3 letters called the anticodon. Not the codon, but this is the anticodon. And an anticodon of tRNA complements a codon of mRNA. I'm going to explain this. Don't worry. But the anticodon on tRNA determines which amino acid it can bring over to the ribosome. So depending on the anticodon, right, that will determine which codon it can recognize, which will specify which amino acid should be inserted into the protein. Anticodon is always written 3' to 5'. So by looking at an anticodon, you can figure out the codon very simply. So I'm going to just suppose that my anticodon is 3' GUC 5'. What mRNA codon would that recognize? So if this is my mRNA molecule here, what 3 letters of mRNA would this complement? Well, it would be C with G, A with U, and then G with the C. So this anticodon will complement the codon 5' CAG. Let me show you that on the next slide. We said our anticodon was GUC. That means our codon must be this. Once we figure out the codon, we can then look at the codon table, right? This is a codon table. So the codon CAG, let's go CAG, encodes for the amino acid glutamine. That means a tRNA with the anticodon 3' GUC will carry the amino acid glutamine. You might be scratching your head right now. If this is the first time you're learning this, it should not make sense on the first time. It requires lots of review, and especially with translation, it's a very animated visual process, so you have to rewatch the animations over and over again until it clicks. For right now, I want you to understand that if the bottom of a tRNA has an anticodon, GUC, it will recognize the codon of mRNA that's CAG. And you could look at a codon table to figure out what amino acid that corresponds to. So we'll talk about this again. So we said the anticodon is complementary to the codon found of mRNA. So if the anticodon on tRNA is AGC, it can only recognize the codon 5' UCG 3' on mRNA. So if this is the codon of mRNA, what amino acid is this tRNA carrying? Right? How would you figure out what amino acid this tRNA is carrying? Well, you would have to look at a codon table. You would look up 5' UCG, right? And you would figure out that 5' UCG encodes for serine, which means that this tRNA must be carrying a serine. So the anticodon complements a codon, and the codon we can look up in a codon table to determine which amino acid that specifies. So let's try to do this together. If an anticodon on tRNA is 3' UGG 5', then what will it recognize on an mRNA transcript? So pause here, and the answer is B, right? If the tRNA looks like this, the UGG, that's like a weird tRNA molecule, and this is my amino acid that it's holding, then what can it recognize on the mRNA? Remember, mRNA is 5' to 3'. So here's my mRNA sequence, ... What is it going to recognize? It can only recognize 5' A, C, C. So the answer is B. And since ACC, right, I could just look up ACC in a codon table and figure out what amino acid this is. Right, and it turns out that ACC encodes for threonine, the amino acid threonine. So let's do a different kind of problem. Let's say I'm telling you that the codon on mRNA is 5' UGG 3'. What amino acid does it code for? So this is the easiest type of problem because you're given mRNA written 5' to 3'. This whole codon table is written as 5' to 3' mRNA codons. Right? So if the codon on mRNA is 5' UGG 3', what does it encode for? You look at the first base U, second base G, third base is G. That means it encodes for the amino acid tryptophan. Right, so a codon table is very easy to look up codons. But if you're given an anticodon that's written 3' to 5', you can't use a codon table. Right? This is a codon table, not an anticodon table. So to figure out what amino acid would be carried by a tRNA with this anticodon, let's figure out what codon the tRNA would recognize. Right? So the tRNA has 3' UGG 5'. That means it would recognize the codon 5' A, C, C 3'. Since this is an mRNA codon written 5' to 3', I can then look this up in a codon table. So 5' A, C, C encodes for threonine. So that means the tRNA molecule that has UGG is carrying the amino acid threonine, because it could recognize the codon A, C, C. Right? So again, if that doesn't make sense, do not move forward. Make sure you understand the connection between a tRNA anticodon, an mRNA codon, and an amino acid. That is very important for you to understand before moving forward. One more rapid response question. So if the anticodon on tRNA is 3' AGA 5', what amino acid would it carry? So pause here and do this out. So we could draw it out. If the anticodon of the tRNA has AGA, that means it could recognize the mRNA codon U, C, U. And what do we have to do? Look at the codon table for UCU. So U, C, U is serine. That means this has to be a serine amino acid. So now let's talk about the steps to translation. We have mRNA, tRNA, and ribosomes all coming together, and we have the same 3 steps as in transcription. We have initiation, elongation, and termination. Let's start with initiation. So during initiation, the leader sequence at the 5' end of the mRNA transcript bonds with the small ribosomal subunit. Then we have the first tRNA comes in and recognizes the start codon. The first AUG of the mRNA transcript is the start codon. So zooming in, the first initiator tRNA binds in the middle of the ribosome at the start codon of mRNA. Right? The first tRNA is always carrying over a methionine. This methionine doesn't always make it into the final protein. Sometimes it gets chopped off. But since the start codon of every single protein is AUG, the initiator tRNA always has a methionine. Once that initiator tRNA is attached, then the large ribosomal subunit can come up and form what we call the initiation complex. Right? So this is the end of, we have this kind of sandwich. We have the mRNA, which has the message. We have our first tRNA at the start codon, and we have our ribosome as like the bread of the sandwich, with the large ribosomal subunit and the small ribosomal subunit, almost like a workbench for the mRNA to lay down on. Right? And remember that the start codon is AUG, so a tRNA carrying the methionine is the first amino acid to approach. So here we are again. There's a little leader sequence before the first AUG. This is the start codon. The initiator tRNA recognizes this, and then a large ribosomal subunit could come and start initiation. We then have the next step is elongation. And what happens is, first, a new tRNA molecule. The first one was already methionine. Recognize the first one. But a new tRNA molecule will then bind to the exposed codon next to the first tRNA. So here's the first tRNA. So this next tRNA molecule will recognize whatever these 3 letters of mRNA is. So the next codon will be recognized. A tRNA molecule will bind, depending on, right, whatever the 3 letters of mRNA are here, that will determine which type of tRNA can recognize it and which amino acid it would bring over. Once that next tRNA comes in, the ribosome will form a peptide bond between the amino acids. The ribosome will then pull the mRNA strand the length of 1 codon. So what that means is this whole thing will be moved over to these 2, will be moved over to these 2 positions. So this part will be empty, and I'll show you that in the next in the next animation I show you. So again, just to recap, in elongation, we have the next tRNA comes into the empty site, and then a peptide bond is formed between the first amino acid on that first tRNA and the second amino acid on that second tRNA. Then both of these guys are going to move over here. These 2 are going to move over to here, and then this will be an empty vacancy for another tRNA molecule to come in. So here's that vacancy. So another tRNA molecule can come in. Again, depending on the 3 letters of the codon, that will determine which amino acid it brings over. Then a peptide bond is formed between the amino acids on this middle tRNA and the new the newcomer, the new tRNA. So this is the peptide bond that gets formed. And then everything slides over 2 places. Right? These two slide over to these 2 positions. And what happens is this tRNA will then be eliminated. It'll exit. And then another new tRNA molecule will be coming into this exposed site. Again, it's a very visual process that has to be watched in an animation usually before it clicks. But pause here and make sure that this makes sense at least a little bit before you move forward. Right. So here we have the last step called termination. An elongation halts at a stop codon. Right? So it's UAA, UAG, or UGA. They do not code for an amino acid. And a special protein called a release factor binds to the stop codon. Not really a tRNA molecule. It's called the release factor protein. And this prompts translation to end. The ribosome will release the tRNA. The ribosomal subunits will then separate, and the polypeptide is released. So here is our finished polypeptide of amino acids connected by peptide bonds. The ribosome can now go on and work on a different polypeptide. This sequence of amino acids could then be folded into a fully functional protein. So I'd like you to watch this animation over as many times as you need to.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites.
You don't need to know the
During
names of those
elongation,
sites.
individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
So again, you can rewatch that as many times as you want to understand the visual process of translation. So to summarize protein synthesis in eukaryotes, we start in the nucleus, where DNA in the nucleus serves as a template, has instructions on how to make a certain mRNA molecule. And RNA polymerase rewrites the DNA message into messenger RNA. mRNA is processed. That has to be spliced all the introns have to be removed, and a special cap and tail have to be put on before the mRNA can leave the nucleus. Once the mRNA leaves the nucleus into the cytoplasm, it could be associated with ribosomes. tRNAs bring over amino acids, and they basically match up their anticodon with the codons of mRNA and put amino acids in the right sequence. And ribosomes put peptide bonds in between all those amino acids. When a stop codon is reached, the ribosome detaches and the protein is finished being made. The ribosome, as it's building proteins, I didn't mention this before, but it can become attached to the rough endoplasmic reticulum. And the amino acids, once they're attached, they can be snaked through inside the endoplasmic reticulum as it's being made. So as this ribosome is being built, as the ribosome is assembling amino acids together, that amino acid chain can be translocated inside the endoplasmic reticulum, where it can be folded into a functional shape. There are special proteins called chaperones that make sure the protein folds into its proper shape. And also there are some post-translational modifications that have to occur. So sometimes proteins need to be chopped at certain places or need to have a certain lipid added or sugar added. So all of that can happen after translation. So watch this video that's contained in the ebook that talks about translation step by step. I like this because you could see, so here's one tRNA, then you have a peptide bond being formed, and everything slides down too. A new tRNA comes in, peptide bond forms, everything slides down. New tRNA, peptide bond slides down. Right? This is showing, again, the process of translation, step by step. So finally, I'd like you to watch this one video that summarizes everything from gene to protein, and this is one of the best videos I could find. I included this on Blackboard, so please watch this as many times as you need to.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So translation is a very efficient process. Some cells of the human immune system can make 2,000 proteins per second, right? That's really important if we want our body to respond quickly to an infection. How do they do that? So sometimes multiple ribosomes can attach to a single mRNA molecule at once, so you can make many molecules of protein from just one transcript. So this ribosome over here started down here, and snaked along as this protein was made. But then this one started shortly after. Then this one started, then this one. So in this way, multiple ribosomes at once can make many proteins. This is called polyribosomes. Another way is that multiple mRNA transcripts can be transcribed, and multiple different mRNA transcripts can be translated from simultaneously. Right? So it's not only that dozens of ribosomes can translate one mRNA molecule, but there could be many mRNA molecules, giving the cell more than one recipe for the ribosomes to read. Right? And like I mentioned, a big part of translation is protein folding afterward, and special chaperone proteins in the cytosol and the rough endoplasmic reticulum ensure that proteins fold properly. Errors in protein folding can occur even if the genetic sequence is unchanged, right? So Alzheimer's disease, for example, is associated with misfolding of proteins and the formation of plaques of certain proteins. They aggregate when they should not. Antibiotics and some poisons work by inhibiting the translation of proteins. So antibiotics work by inhibiting bacterial translation. So for example, penicillin and chloramphenicol work because when we take them, it prevents bacteria from doing translation. But we can still do translation. If bacteria can't do translation, that means they can't make any proteins they need to survive. Right? So that's why antibiotics work against bacteria and not for us. It only affects bacterial translation and not human eukaryotic translation. So you can pause here for a summary. And here is a practice problem that we'll do together during recitation, but I'd like you to do this now to get a sense of it. So if you're given a template strand from 3' to 5', you're first going to be asked to write the mRNA that would result after transcription. So what would you get from this? This is the DNA template. What would your mRNA look like? From there, you would have to break down the mRNA into 3-letter codons, right? Every 3 letters of mRNA is a codon. So then write down each of those codons here. And then I want you to look at a codon table to figure out what each codon encodes for, which amino acid is encoded by each codon. And the reason why I want to show you this is because sickle cell anemia is a very good example of how just one change in the DNA code can affect the entire protein. Right? And we'll talk more about this next. So I want you to look at this sequence for hemoglobin. This is a portion of the genetic code for hemoglobin, one of the proteins in our blood cells. And the 6th codon is usually GAG, which encodes for a glutamic acid amino acid. So the DNA would usually have CTC, and that would encode for the amino acid or the codon GAG, which would be the amino acid glutamic acid. However, suppose I have a mutation in my DNA. There's a typo. And instead of CTC, the DNA has CAC. Well, that one single mutation would cause a different codon to be put in, right? It would be GUG instead of GAG. And GUG does not encode for glutamic acid, it encodes for valine, an amino acid with very different properties. Because of that one substitution, the entire hemoglobin protein misfolds and aggregates. They stick to each other. And that causes the red blood cells themselves to have a sickled shape. And because of the sickled shape of red blood cells, they cause anemia. They cannot deliver oxygen efficiently throughout the body. So this is a very good example of how one single letter of DNA could cause a change in the amino acid sequence, which can change the protein folding, which could change the structure of the cell, which could change the functioning of that cell, right? Red blood cells can't do what they normally are supposed to do because of sickle cell anemia. So that is the end of the first part of Chapter 7. Please watch translation videos as many times as you need to before moving forward. Next, we'll discuss gene expression.